So all welcome in behalf of the Maastricht University. My name is Jean Mures. I'm a professor of general practice and I will be your pro-rector. I will take the English version. And hereby I open this academic ceremony in which Vinidhra Shankar will defend the academic thesis, Well. What have we grown here? Scaling up bioengineered embryo models for biomedical applications. May I invite you, dear Mrs. Candidate, to present the summary of your study in layman language and the conclusions of your thesis.
Dear Pro-Rector, thank you for the kind introduction. A warm welcome to my defense committee, supervisors, family, friends, and colleagues present here today. For the next 15 minutes, I will be presenting the key findings for my PhD thesis titled, Well, what have we grown here? Scaling up bioengineered embryo models for biomedical applications. Even before birth, a baby's health can be shaped by the mother's environment. A growing fetus inside the mother's uterus is highly fragile, especially during the early stages. Any disturbances during pregnancy could affect normal embryonic development. For example, pharmaceutical drugs that are prescribed for pregnant women should be tested for their effect on the growing embryo. In parallel, exposure to certain chemicals, infections, and habits of the mother can disrupt normal fetal development, causing birth defects or congenital abnormalities. Hence, the study of developmental toxicity is of clinical importance. Until now, developmental toxicity tests have been predominantly studied using animal models. This is because of two reasons. Firstly, the conduct of experimentations with human subjects or human embryos poses a variety of ethical concerns. And secondly, many developmental pathways are highly conserved across different mammalian species, especially between mice and human. So animal data can give valuable warnings about potential human toxicants. However, research involving mice faces considerable limitations in sustainability, feasibility, and scalability. Importantly, millions of animals are sacrificed per year in studies of toxicity testing in order to approve a drug. With the current climate around minimizing animal experimentations, and the recent FDA guidelines for implementing the 3 Rs, reduce, refine, and replace, there is an increased need for developing stem cell-based models that can allow developmental toxicity testing. In order to study developmental toxicity, we need models that can help us recapitulate development in the lab. Mouse embryonic development is a complex process, controlled in space and time. Stem cells can be derived from the embryonic tissues, which can then be used as a starting point for modeling development in a dish. The resulting structures are called stem cell-based embryo models. Recently, there has been extensive research in the development of stem cell-based embryo models for mice and human. Here, the stem cells are directed to differentiate into structures resembling features or aspects of natural embryos. These models can be a powerful system to not only promote the fundamental understanding of development, but also to advance reproductive and regenerative therapies. Despite their utility, embryo models can display limited complexity, throughput, automation compatibility, and the extraction of real-time morphological readouts. Further, there is a lack of automated tools to measure and quantify development and its associated stages, thereby limiting the translation of these embryo models to screening domains. Hence, the aim of my PhD was to develop a scalable, robust stem cell-based embryo model compatible with high-throughput screening and developmental tracking. Our first step was to develop a high-throughput phenotypic screening and analysis platform. This required a versatile system that simultaneously allows the reproducible and long-term culture of structures with precise control over the cell numbers. Further, they should also allow in-plate imaging and automation compatibility. Hence, we chose a microwell-based platform which involved a 96-well plate where each well contained several small engineered microwells inside that are designed to provide a controlled microenvironment for cell culture, thereby allowing the controlled self-organization of stem cells in a high-throughput manner. With the platform chosen, the second step was to develop a stem cell-based embryo model that can faithfully recapitulate aspects of mouse embryonic development. We started by seeding mouse embryonic stem cells into the microwells and cultured them for 5 days. Upon providing chemical cues, the stem cells self-organized into a structure resembling a partial 5.5-day mouse embryo. One unique feature of our embryo model is that the different stages of its development inside the microwells closely resembled the sequential stages of natural mouse development, hence offering an elegant and relatively simplified yet powerful model for developmental toxicity screening. The most significant advantage of this platform is its scalability, enabling the generation of hundreds to thousands of structures per plate in a high efficiency. The structures can then be imaged within the microwells and used for further downstream image analysis. Which brings me to the fourth and the most important feature of our system, the automated image analysis pipeline. To analyze the large number of image data sets obtained, we created a pipeline using a software to extract different morphological parameters, such as area, size, shape, intensity, radial intensity, eccentricity, and more from these structures. Using this data, we could then classify the different morphologies of structures observed per well using supervised machine learning inbuilt in the software. And further, also measure the yield or ratio of the different classified morphologies. This measurement of yield can shift when the embryo model is, for example, treated with a toxic compound. And this way, we can generate a large number of image-based automated yield measurements, which can be used to study the effect of a toxic compound on the development of our embryo model. Coming back to the initial motivation, we then wanted to use this automated image analysis platform and our embryo model to perform a high-throughput developmental toxicity screen of compounds affecting development. We use the same protocol to form our model, but between day 2 and day 3, we expose the cells to a library of compounds. We chose a diverse library of 30 compounds consisting of known teratogens or known compounds affecting development such as thalidomide, commonly prescribed drugs such as aspirin, vitamins, caffeine, pesticides, and more. After 5 days of culture, the resulting structures were imaged within the microwells and analyzed using the automated image analysis pipeline. Based on the morphologies observed in different treatment conditions, the yield measurements were obtained. This is a graph displaying the yield measurements after treatment with compounds at 5 increasing doses. We observed that the higher doses of the compounds, indicated by the red arrows, imparted a highly toxic effect on the embryo model, correlated by the significantly reduced yields. Through the yield measurements, we were able to achieve results comparable to studies using other mouse models. One interesting finding from these measurements was the exposure to a compound that is very well known to all of us, caffeine. There has recently been some studies using mouse embryos that showed a developmental effect upon exposure to high doses of caffeine during the very early stages. Alluding to that, in our model, we see an interesting morphological effect that could possibly be attributed to the effect of caffeine on a crucial developmental pathway. Although caffeine is not toxic in general for adults, the case is different for an embryo. Our pipeline was able to pick up these morphological changes due to compound exposure. Hence, through this study, we also wanted to emphasize that traditional cytotoxicity assays alone insufficiently predict the effect of a drug on the development of the embryo. Hence, we show that through our automated image analysis pipeline, we can efficiently detect morphologies due to compound treatment. And we propose that they can be used to complement traditional cytotoxicity studies in order to predict toxicants better. Now, the embryo model we had developed recapitulates the stage of embryo during the implantation. With the potential of this embryo model and the microbial-based platform that we had developed, we wanted to push the boundaries of the system to allow long-term culture of these structures beyond the implantation stage. Interestingly, when we cultured the day 5 embryo model for 2 weeks, they spontaneously self-organized to form complex tissues that were reminiscent of an embryonic yolk sac. On day 10, the mouse yolk sac is essential for embryonic growth, acting as a selective barrier for nutrient, oxygen, and waste transfer. In our model, the yolk sac-like structures gradually expanded to develop beating cardiac tissues around day 10, which is also the first organ to form in a mouse embryo, and later showed reminiscence of blood island formation on day 14. As pictures speak louder than words, this is how the structures look like after 14 days in culture. Here, we are looking at the yolk sac structures encasing a cardiac niche that is beating and showing signs of early vasculogenesis and potential blood island formation. Through our culture, we were able to successfully direct the stem cells to self-organize from implantation stage to early organogenesis. And then we use this yolk sac-like model to track development. As per the theme of my thesis, we developed a custom microwell-based platform that can host the development of multiple yolk sac-like structures from day 0 until the entirety of its development. The microwell-based platform allowed us to track the continuous development of these yolk sac-like structures over time. In the future, this system can allow high-throughput toxicity screens to study the long-term effect of drugs on development, and also to perform screens to modulate developmental events leading to organogenesis. With that, I would like to summarize the main goals of this thesis. I started by developing an automated phenotypic screening and analysis platform, and in combination with our stem cell-based embryo model, we performed a high-throughput developmental toxicity screen. Further, I used another microwell-based platform to direct the differentiation of this embryo model towards organogenesis to track development over time. As a final conclusion in this thesis, we developed novel microwell-based platforms for diverse biomedical applications. We are at a stage where the shift towards non-animal alternative models are inevitable, and the need for next-generation analysis tools to extract and validate the large number of data sets is ever so high. And I hope through this thesis, we have played a small part towards this. With that, I would like to thank my supervisory team and the whole Merlin team for helping me with the project, and to all the funding bodies, and to you for listening. With that, I would like to give the word back to the prorector.
Thank you very much, Mrs. Candidate. If you would go back to your previous slide about your conclusion. So you have 2 conclusions. Could you translate those conclusions, who are in academic wording, into a layman summary? So what you say to your family, what your conclusions are?
So I have created a platform that can help us study the developmental toxicity studies, or toxicity tests, using our embryo model in a more scalable manner. And this can also help in using a model that is not involving animals. So by reducing the use of animals for these studies, we wanted to present a novel study that can then translate these complex developmental stages into a model that can help studies of toxicity better. So hopefully this is a start to a whole generation of these in vitro models using new approach methods to then push the field further towards the toxicity tests.
Thank you for your extended presentation. And the opposition will be opened by Professor van Griensven. His chair is on regenerative medicine. Professor van Griensven.
Thank you, Mr. Prorector. Dear candidate, how much I love to read your book, and then when I got it in my hand to see the nice pictures in the front, it was really nice. Also, of course, the supervisory team, my congratulations. I would like to start with you to speak about chapter 5. So in the abstract, you state actually that you showed a multi-organ vascularized embryo model. Did you really show organs?
To your highly esteemed opponent, thank you for your kind words and for your question. We did not show, what we see from these structures are initial progenitors of heart and cardiomyocytes, but also signs of vasculogenesis, which we then confirmed using other stainings and single-cell RNA sequencing, which did show markers for these structures present for these tissues. What I would say that they are a starting stage to the organ formation. They are still in tissues, and this is how the yolk sac in the in vivo stages also form, with this formation of these yolk sac, which then is the site for preliminary or primitive hematopoiesis in the mouse yolk sac, and that later starts to form these blood islands and vasculogenesis, which happens also sequentially. So through our model, we were able to show the initial signs of these structures and tissues present, which, yeah.
Okay, to further discuss with you, I would like first one of the paranymphs to read proposition 9.
Your scientists were so preoccupied with whether or not they could that they didn't stop to think if they should.
So, did you stop thinking?
Yes, I did. Definitely. I mean, that's also something it's a key thing that we as scientists in this field, especially in the field of embryology, need to think once in a while, and something that the whole community of the ISSCR guidelines also strive to think, because right now there are certain guidelines which we as scientists should not proceed beyond, and there are checkpoints where we should be aware. So as a short answer, I did think about it.
Were you at the checkpoints, or were you still before the checkpoints?
Before. Yeah, also to say that these are mouse models.
Right.
And one interesting thing is that there are two types, which are integrated and non-integrated models. And ours is a non-integrated model, which means they do not have all the essential tissues that are required for the formation of the embryo. So they only represent certain aspects or features of a natural embryo. So we don't form these structures. So we have that is also a checkpoint with our model.
Yeah. That's interesting. So taking it like the ideas, what you said in the beginning, and now also your answer, I still am a bit puzzled with the organ thingy. So you did bulk RNA sequencing, and as I understood correctly, you made it from the beating structures, right? So, and then I see cell types such as gut, forebrain, etc. How do they come in your beating structures?
Yes. So there. The thing about these embryonic development is some of the initials, so they form during gastrulation, they form 3 germ layers, the endoderm, mesoderm, and ectoderm. And even though most of our structures, or most of the cell types that are formed in these structures, go via the mesendoderm, so mesoderm plus endodermal lineage, there are these other cell types that do emerge in a minority fraction, but they do emerge. And that can be falsely translated to forming neural tissues, but they are indeed in the very early stage, which because of the sulfate, like the combined correlated sulfate markers that are there for these specialized tissues that could be coinciding with ectodermal tissues. So we don't necessarily see the other. Also for the gut is also an endodermal tissue. And even though we form more earlier stage, the markers that are for gut endoderm also coincide with the markers that are there for the earlier endodermal stages. So it also does something with the annotations of these during the bulk sequencing.
And do you think bulk sequencing is then the correct method to use, or?
No. So yes, it is also the first thing to do because we were looking at structures which form these kind of phenotypes, but so we wanted to know what exactly we are looking at. Usually, bulk sequencing provides some answers to these questions. Okay, as a bulk, what exactly we are looking at, and once we were sure of what we are interested in, we then did a single-cell RNA sequencing for these breeding structures to then see more into detail on these phenotypes. So bulk did provide us some overview of the cell types and the markers that are present in these models.
And when you then look at those signatures you found, is it then similar to the signatures that are found in a real embryo?
Yes, I would believe so, because when we did the bulk sequencing, we also overlapped it with the natural mouse embryonic database that is publicly available, the repository, and we then overlaid it with the genes that are there in the real embryo to what we see in our model. So we did compare the model's expression patterns to that was available with the public repository on the database.
Nice. And then you also see actually a lot of CD31 positive structures, right? So you have this whole vascular network, but actually you have very nice pictures in there. Do you think that these structures are more like from the organs, or is it also more that would, like in a real embryo, more go towards the placenta and make the connection with the mother?
So that's a very interesting question. So the CD31, after we did the stainings as well, what we saw was that these blood vessel-like vasculogenesis that was happening was always around the structure. So if you consider so mostly around enveloping the yolk sac,
exactly,
which is also what we see in that is the connection between giving the transfer of nutrients and acting as a barrier. So that is also what we see in the real embryos. So we did see a network that was completely surrounding these structures. It was mostly on the surface.
Mostly on the surface, okay. I think my 9 minutes are over. Thank you very much. I'm happy with the answers, and I give back to the pro-rector.
Thank you.
You could have had 2 minutes, but now it's gone. Professor de Boer will take over from you. He is a professor of biomedical engineering at the Technical University of Eindhoven. Welcome after such a long journey.
Thank you very much. It was a very long hour. Congratulations with the completion of your thesis. I would also like to congratulate the supervising committee. I read the thesis with a lot of enthusiasm, and I've definitely 9 + 2 minutes to discuss with you. So the first question I would like to ask you is about chapter 5, in which you say that there's development of a yolk sac. So there's early hematopoiesis ongoing there. And I know that in the field of blood transfusion, a long-term repopulating hematopoietic stem cell is sort of the holy grail that everyone is after. Do you have it? Do you have a long-term repopulating cell in your tissue? Well, in the tissue of your embryos then.
To highly esteemed opponent, thank you for your kind words and for your question. That is indeed a very interesting and exciting question. So in our mouse model that we have, we do see blood islands, which are clearly red under the microscope in a general bright field. And when we then did flow cytometry to then analyze for the different progenitor markers panel that is also observed in mouse primitive blood, we did see some populations of EMP, so erythrocyte, megakaryocyte progenitors, and also some in the CMPs, which are also myeloid progenitors. So we did see that, which we also confirmed with SEM, so scanning electron microscopy, where we did see not just primitive erythrocytes, which were which you can see under the SEM microscope. But we also see around these structures, around these erythrocytes, there were macrophages, early macrophages.
Okay, so you do have evidence.
Yes.
From your in vitro model that there are things that may be hematopoietic stem cells.
Yeah.
So, do you think about a potential experiment to actually prove that you have long-term repopulating cells?
Yes.
What would you do?
Yes. Thank you for the question. So yes, that would be definitely the next step we would want to test. So if we can isolate specifically these populations using FACS or just laser cutting and then differentiate them in vitro to see if we can then differentiate these populations towards more mature erythrocytes or mature cell types, then that would be a really nice study to see that they have the potential.
That's not the definition of long-term repopulation in the hematopoietic field.
Sorry?
That's not the definition of long-term repopulation in the hematopoietic field. Have you thought about functional assays that you could do with your cells in order to prove your functionality?
Yes. So I did not have the time to do those assays, but I did think about the functionality. It would be nice to check if these cells could then act as a separate source to produce more cells of their own.
No, it's not really the direction I'm thinking about. I'm actually thinking about lethally irradiating mice and then injecting cells in the mice to see whether they can actually repopulate the hematopoietic niche. That would be the ultimate proof of your. But I was just wondering whether in your group you have discussed this and whether this is. I mean, is your technique at the moment mature enough to be able to do such an experiment?
Not yet. So right now they form these.
So what are the bottlenecks then?
Sorry?
What are the bottlenecks? Why could you?
So right now they are forming these primitive states. We are yet to confirm if they can actually mature to form the experiment that I mentioned earlier, to form actually the mature erythrocytes. If they can still, and also to differentiate into other blood cell types. If these niches can indeed do that in the lab, then we can already think that, okay, they have the potential to give rise to more mature, because they need to mature and move towards, from the yolk sac, they need to go also to the bone marrow, from which they need to migrate. So hopefully we need to be able to do that.
Okay, thank you. Another question I have is about the use of the pseudo-embryos as a source to test toxic compounds. So you're now using them, you expose the embryos to those compounds, and then you measure things, cells dying or developmental defects. But you're looking at the features that you're measuring or the things that you're measuring are in the embryos. Whereas I think that usually developmental toxicology is not really evident in the embryo in the pregnant woman, but it's actually evident from the newborn. Mm-hmm. So that's much later. So how relevant is it to actually measure these things in the embryo relative to being able to measure it in the newborn?
Yes.
So is your system maybe a little bit not too early? Because actually what you're interested in is the effect on the newborns.
Yes. So these studies, so what the problem with also the human embryos, or just embryos in general, is we've not been able to do these tests so long because of that reason, that they cannot be accessed. So what we can do with the live embryos is you can inject them, and the only readout you can get is after. And that is why we don't have any data on how it affects through the stages, which would already be a good indication of why is it happening, instead of waiting for months to get an actual result.
You're saying that...
So these models can actually, so there are several other models which recapitulate different windows of development, and that can give us clues on why this is happening. Maybe, of course, these models will not replace a live embryo.
That's a good point. So you're saying that if you have known teratogens, at least you can use the models to see what is happening in an early stage. But I think it's a little bit more difficult to use it as a method to find new teratogenic compounds. But if you would, how would you do that? I mean, what kind of features do you have to extract from an embryo to be able to say that later, you know, in a newborn stage, you may have an effect?
Yes. So the stage of embryo that we are also looking here is the implantation window, which is also a key developmental stage that can affect future developmental events. So this is a stage where major cell fate decisions happen, the formation of the luminogenesis, which then leads to gastrulation. So those are the stages that do indeed give rise to the organs later. So the formation of the 3 germ layers. So if we can identify that these compounds are, even if there are new teratogens that can affect these compounds, these stages, then we have an indication that, okay, this compound at this stage could be affecting the formation of a mesosome. So then when you do the studies later, you can still confirm, but this will be an initial screening step. Yeah. So before you do a major 1,000 molecules on millions of animals, this will be an initial screening steps to already see which stage we can actually target with these. And also to create these kind of experiments, we need clear outcomes. So if we are interested in so if we are creating new drugs and we want to see which stage of embryo we predict that it would affect, we can use a specific model and then do an initial screening step. And then with that data, with the confirmation with the mouse data, then we can move to the next stage.
Yeah. Do we have time for another question? Yeah. So let's follow up on this. I think this is interesting. So it means, how do you get as much information as you can from a model? And in your thesis, you've been using feature extraction. So you used, I think, Cell Profiler to extract features. Did you also consider, or would you consider using other methods to basically featureize the embryos that you're working in? Did you consider, for instance, deep learning type of algorithms instead of feature extraction? And how would that be implemented? How would you train a deep learning model on the type of screens that you perform?
Yeah, absolutely. So that is also something that, this is a proof of concept study that we wanted to test with, because here we extract, so we don't directly extract information from images, we extract morphological parameters, and using those morphological parameters that we've extracted, we then use the machine learning pipeline to do the classification. Yeah. But with deep learning, we can then use this image data, the vast majority of image data that we have, to then directly predict how can we classify the different morphologies observed, and how can we...
But how would you train your classification model?
So the initial step we should do is to have clear outcomes. So having true positives, true negatives, and also, yeah, that's something that can clearly show which phenotypes that can actually form through these exposures, and then classify and do the training for the deep learning algorithm. Yeah. And once you get that, I think can...
I'm looking at the chairman. A small question?
No small question. No. You can...
Very much for your clear answers. I will give the word back to the director.
There will be discussion afterwards with the candidate, I think. The opposition will be continued by Dr. Morris. Her expertise is also in developmental models at the Francis Crick Institute in London. Dr. Morris.
Thank you. So I will start with a more philosophical question to kick us off. You've used the term integrated and non-integrated in your presentation and in your answers to my colleagues. I wonder whether you have any thoughts on the use of that term. I know there's been a lot of discussion in the field, and whether you feel it's a useful terminology at the moment.
Dear highly esteemed opponent, thank you for your kind words and question. So this... Yeah, it is a very crude definition, integrated and non-integrated, that we are trying to box what exactly, like having a full-grown structure that completely recapitulates natural development, or just aspects. I do think it is a valid classification to start with, to see, okay, if we form, like for models, for example, like blastoids, which actually formed all the 3 different tissues that we see in a natural blastocyst, which can then have a more higher potential to then develop, and the closeness that it has to the natural embryo, and calling them integrated, yes, I think that would still be key, because then we can have the guidelines that are required for, so I think the recent guidelines for ISSE are also use different regulations to then check on these models based on what features they develop. So having an integrated model makes it a higher priority to be checked for and what kind of timelines they form. But so I think it does help.
The ISSCR 2025 revision recommended moving away from integrated and non-integrated for precisely that reason, that they weren't capturing the kind of ethical considerations that might be part of an embryo model. So it's interesting that you stand by it still.
No, it is so for now it is a crude definition, yes, but I do think there would be a time when we need to really define where exactly we need to have these checkpoints, what exactly the stages they are forming in tissues. I think that's the next step, definitely, in moving or creating routes for these embryo model studies.
Okay, we'll move on to some biology then. And I wanted to pick up on these models that have the yolk sac fate. It wasn't completely clear to me, but it was my understanding that you see these epiblast-derived cells, like the cardiomyocytes, inside of the yolk sac structure. And I wonder how you explain that in light of mouse embryology, where, of course, the yolk sac doesn't surround the embryo.
Yes, that is very true. These are also, so in our model, we chemically induce both the extraembryonic endoderm and the epiblast lineage. So they co-develop from the beginning to envelop the epiblast. And when they further expand, we do see these morphologies. Of course, in the true embryonic development, the yolk sac sort of goes around it in a fluid-filled sac where all the three germ layers with the embryo proper goes inside it. But that is a key difference, I think, in our model with the embryo model as well.
You don't see, for example, an amnion, which would be expected to cover the embryo.
Yeah, no. In this current model that we have, we don't. Okay. We did test for in the single cell sequencing, which did pick up on some genes, but physiologically we don't see any amnion.
Okay, and then I wanted to pick up on the epiblast-derived cells in particular. So you've described and you've shown us videos of these beating cardiomyocyte structures, which of course are derived from the anterior end of the epiblast. So I wonder whether you see, let me rephrase that. I wonder to what extent you see organization in the epiblast cells. Do you see an anterior-posterior polarity? Are the cardiomyocytes organized in any way into a tube or a structure?
Unfortunately, there is a lot of heterogeneity in the cardiomyocytes that form in these yolk sac-like structures. We don't necessarily see an anterior-posterior polarity here. So what we form are beating cardiomyocytes that are somewhat scattered inside these yolk sac structures, but they don't always polarize to one side. We don't. Sometimes the whole structure also beats. So they could be,
Okay, and then I'll pick up on that point about the heterogeneities, because I think that's really important for your signal modulation, where if you're obviously varying the degree of nodal or the degree of Wnt, that's going to bias the cells more anterior or posterior. How do you interpret that in light of the variability of the system?
So that is a great question. When we did the bulk sequencing, we also saw that the structures which did not form these yolk sac-like structures. So there were other structures which were left behind that did show more ectodermal or neural markers. So that could be also due to the kind of paracrine signaling that the cells are getting from each other, and secondly also with the cell number. So some clusters form bigger cell number, like with higher cell number, which then promotes the differentiation of these cells towards a mesendodermal lineage, thereby forming these yolk sac structures. However, we do see in the heterogeneity point of view, we do see some structures which do stay back and differentiate internally without increasing in cell number or having any structural organization. So they showed more of an anterior cell types. So that did occur. So these kind of, so that's why in one of the later stages of these experiments, we did try some pathway modulation. So this was the model that we got initially, but then we did, using the microbial-based platform, we could then test for different pathway modulators. And as you rightly pointed out, when we added BMB4 to a compound, that did boost the yields of formation of these yolk sac structures. And when we added XAB, which is a Wnt inhibitor, they did form lesser of these yolk sac-like structures. So there is definitely a key, the modulation of the pathways plays a key role in formation of these structures. And that's, I think, would be the next step to test, to have like a panel to test different modulators at different time points even, to see where, if we can boost the cell type towards a specific tissue organization.
Mm-hmm. Okay, so on the similar line, but moving on to your vascularization, I think you've shown convincingly that you have endothelial cells based on the flow cytometry data and some of the stainings and qPCR. To what extent is this a plexus, or is it really the formation of tubular structures that would be sort of required for vascularization, the sort of classical definitions of vascularization?
Yeah, thank you for pointing that out. So we did indeed see initial signs of the formation of these progenitor markers, but with the tubules that we got, or the structures that look like vascular structures under the microscope, we were not able to, we did not have the time to study the functionality of these structural vascular tissues to see if they have a tubular configuration. That would definitely be the next step, to also study to see if these vascular structures do have any, the tubular nature to have them flow through something. So we haven't had the time to test it yet.
Okay. Final question. You show that you've got these longer term culture by keeping them in these microfluidic chambers. I wonder why you stopped the experiment at 14 days. Was that biological or technical? And what was the result? What did you see at 14 days that was different at 10 days?
So that is a great point. When we did the experiments, the initial experiment was then flushing them, these structures, to a 6-well plate, where the beating and the yolk sac-like structures formed around day 14. But when we then translated it to a microbial-based platform, they formed these kind of morphologies already within day 11. So could be the confinement of these structures within microwells, and also we saw similar results with the flow conditions, where we also wanted to boost the flow of the oxygen and the media through and the nutrient exchange for these structures. So we stopped the experiments, wanted to stop the experiment earlier because we could see the formation of these structures. I did not have the chance to show it, but we were able to see beating and yolk sac-like structures in these microwells already around day 10, 11. So the results from these experiments are that we saw it, but unfortunately due to time, I didn't have the chance to characterize them further. The idea was to also test the flow versus static condition to see if we could minimize the nutrient depletion and the oxygen exchange with the flow. If we had managed to do that, but that's something we would like to test for sure.
Thank you.
Thank you very much, Doctor. I would like to invite Dr. Schreuter to continue the opposition, and Dr. Schreuter his expertise is on stem cell-based developmental models from Leiden University. Welcome.
Thank you. First of all, I wanted to congratulate the candidate on the hard work that you have done, on the very beautiful data that you've generated, and on the very nice thesis that you have produced. So I will start my questions also with one of the later chapters that I think has intrigued a lot of the opposition committee, the fact that you see so efficient mesoderm differentiation, right? And it's very impressive when you show this in your talk, also these beating structures, but when I saw it, I was surprised. And that is because your structures are different from the embryo, and I think they don't, usually you would expect that they don't really conform to the requirements for mesoderm differentiation. So I wanted to ask you if you could contrast the signaling environment in the embryo with the signaling environment in your structures, and speculate how is it that you do get mesoderm differentiation in your structures.
Dear esteemed opponent, thank you for your kind words and for your question. In our, so as you've rightly pointed out, so our structures do look completely different than how it happens in the natural embryonic stages. Here, the fact that we're also chemically inducing for endoderm and epiblast to start to grow together already puts them in this rosette-like conformation, which then expands further to envelop the embryonic tissues. Although in the natural state, there is also the trophoblast environment, which then also leads to the formation of extraembryonic ectoderm, that then signals for adding the nodal and the BMP morphogen gradient that leads to the formation of the anterior-posterior patterning. Although in our tissues, we don't have that compartment, which is why when we did the signaling pathway screening with the BMP, we could then push them towards more mesodermal lineage. So I think in order to replicate something like what happens in the natural state, it would be important to have like a morphogen gradient. So if we can model somewhere where one side of it could be a BMP high and nodal and BMP low on the other side, maybe then we could have more anterior-posterior tissues. But right now we have, as a whole, we are exposing them as a whole to the signaling chemical induction that we give. So they didn't.
Yeah, I like the idea of gradients. So maybe you can expand on this. How can you envision an experiment or a bioengineering approach to generate such gradients?
Yes, so we did have a lot of discussions about it, even with our team, about using a microfluidic channel where we can then embed these. So already in my chapter 6, we did show that these structures can grow inside a microfluidic platform, but using maybe a gel-based gradient to embed them on into a well, and then through one side, because these are porous microvells as well. So if we can give them a different morphogen on the in one direction of the flow and the opposite direction in another, the antagonists, maybe we can push them towards, give them some kind of gradient on both the sides, and maybe they might, get a mixed signal.
Yeah. Thank you. I will stay with the topic of signaling environment and in the structures that you generated, but going to one of the earlier chapters. So I noticed that in chapter 3, you tested the requirements for different signaling pathways by inhibiting them at different stages. And it seemed to me that there was a temporal switch between the requirement of nodal and BMP signaling. So when you inhibited early nodal signaling early, you got effects, but not when you inhibited it late. And it seemed to me the reverse was true when you did those experiments with BMP inhibitors. And I was curious about your explanation of these temporal requirements.
Absolutely. So the embryonic development during this peri-implantation stage is exposed to a high levels of morphogen gradients, and that's the stage when they also undergo these crucial sulfate decisions. So the timing between, for the epiblast and to then differentiate into, or the inner cell mass to differentiate into epiblast and the primitive endoderm tissues, or formation of these extraembryonic tissue, is already one checkpoint. And that in our model, that was between day 0 to 72. That's when the differentiation towards the epiblast and extraembryonic endoderm occurs. And when we then, so that was, during that time, if we block the MAP signaling pathway, for example, using PD32, we were then able to block the formation of these extraembryonic endoderm tissues. So they then stayed as like an embryoid body mass or an embryoid body aggregate. But then in the later stage, when we then use the same blocking, that did not specifically affect the formation because it had already formed. So the temporal switch is very important because that's the stage when the formation of these extraembryonic endoderm occurs. And so that was also true for the chirin, the Wnt pathway, for example, as well. So when initial stage, there is this rosette-like stage that forms, which then, if we block it during that stage with the Wnt inhibitor, then we can.
So I think these effects of FGF and Wnt are fairly well known. So what is the role of nodal and BMP specifically? What did you learn there?
So the nodal signaling in our model, when we exposed them to the later stage, there was an interesting effect called delamination that occurred, which was something that was also seen when a nodal knockout cell line was used. And I think this is also something, or I speculate that this could be due to disruption in the formation of these rosettes, and during the luminogenesis, when there should be the water gradient that falls into and then creates these polarization, apical basal polarity, there is some disruption due to this nodal signaling that happens in the later stage, which then leads to formation of the lamination. The other speculation I had was also the cell number. So in order to reach a certain, the rosette to the xenepic stage, it needs to have a certain cell number. And the blocking of the pathway during that time also could have blocked the cell proliferation, which then made it still form the xenepic, but then where the size increased, but the cell number didn't increase, so there was a delamination that occurred. So
okay, thank you. If I still have 1 minute, small, yeah, I would like to discuss one of the propositions, proposition number 6. So.
Automated detection and quantification hold great promise for the future, offering an efficient way to streamline the identification of the most relevant and interesting phenotypes in the 3D cellular structures.
Yeah, thank you. So I thought the machine learning approach that you developed was really very innovative and promising, as you write in this proposition. So you have by now used it to form correct xenepic structures, and I wanted to know from you, how do you see this going forward? So what is the next developmental event that you could try to catch with this approach?
That's a very interesting question as well. The reason why we could also do this with these models was, even though they were still, they did have these 3 tissues, they were still simplistic enough to identify the different morphologies that we could then classify with these bins. But the further it grows in the development, as we see with these yolk sac structures, it would get more and more complicated with the formation of different morphologies. That would be a bit harder to then classify effectively with these, with the current pipeline that I have. Then I would have to use more deep learning-based approaches where I can then sequentially track the development and then also use these advanced tools to predict, classify these morphologies. But with the current technique, it might get harder to do it after the stage.
Thank you.
Thank you very much. The opposition will be continued by Dr. Bourognin. Her expertise is mostly on neurodegeneration, I understood. And please.
Thanks a lot, Prorector. Dear candidate, congratulations also from my side. Excellent work, really. Because these days we really need standardization procedures to really push forward this model. So I'll start the questions by picking up a bit on chapter 4. It's difficult to set up a toxicological study, especially in these models. And also because we don't see the outcome of it, we cannot test the entire phases of the embryonic development. But how do we make sure that we are really testing developmental interesting pathways rather than assessing stress or habitat-specific responses on our tissues?
Dear esteemed opponent, thank you for your kind words and for your question. If I understood your question correctly, you asking me if we can classify the different phenotypes due to the teratogens or these compounds rather than the environmental factors such as...
Yeah, or for example, since we are testing, you were testing cell death in most of the assays, yeah. How do we make sure that this is a kind of cell death which is relevant for developmental trajectories?
Yes, so in our model, we do test for at least the cell death part of it by clearly validating with the control conditions. So one key thing we wanted to also establish before we did the whole study was to clearly define the efficiency of formation of these structures in the certain conditions that we have, where there is no cell death that is due to, we also have enough like controls for the DMSO or the solvent-based controls, as well as plate, due to plate effects. So we did account for all of that in the initial stages of the study in order to avoid effects or occurrence of any environmental factors on the development of these structures. So that is a key thing to do before doing a high throughput screen of compounds in order to eliminate any environmental effects or hypoxia or oxygen depletion that happens. So in our studies, I think we did the controls.
And if you check the literature, is data available to benchmark the fact that seeing cell death at that stage would result in this particular phenotype in the mouse embryos, or,
yes,
or not really?
There are sufficient information available with other models as well on what we are supposed to expect in this specific developmental stage, although there are differences between in vitro models and the in vivo system, and between the different models as well. So what we benchmarked against right now was with the control situation, but also with the other in vitro models to see if the compounds also behaved in a similar way with another in vitro model. So then we could have an idea. So there are other models to benchmark against right now. The next step would definitely involve benchmarking with a larger library with the mouse data as well.
Thank you. And if we have to compare a bit the answers we can get from these in vitro models and what we classically have from animal models, because in your presentation you were mentioning that developmental trajectories or pathways are quite conserved between species. But I would argue that actually some, especially for some organs, there are quite different. So if you had to convince regarding the value of your model as alternative to animal models, from a biological perspective, what particular features would you highlight?
So there are a couple of factors that I would highlight with your question. So the model that we have, the in vitro models, they do recapitulate some aspects of the natural embryonic development, although the studies of the embryonic developments usually, or the studies with natural embryos occurs with the different readouts. So they usually have test compounds and then check for specific, in the later stage, more in like a limb development, or if they have a neural development or physiological development at the later stage. So there is not enough studies to test the intermediate stages yet. But that is why these models will also be important to benchmark against the mouse data as well, to see if we can recapitulate if we can have a long-term exposure studies, we can see a similar effect. Also, right now, there is a lot of data on multiple, wide variety of toxicants using mouse models. So I think that can already start as a starting level to then translate to in vitro models as a first step to check if we can actually recapitulate or get similar trend of data as we see in the natural embryos, which can then translate to the human data with human similar in vitro models.
Thank you. And if we talk broadly about development of models, sometimes when we have to keep or ensure high throughput automation, we are often losing on biological relevance, or there is a trade-off. In your particular case, did you have to compromise on biological features or relevance to to obtain high throughput, or not so much?
Biologically, we did not have to have any trade-offs. But we did have to think about the time of, so these are the studies we did are also based on image-based readouts, so which meant we also have to have more automated images and a good quality images that we get without compromising also the time of creating these images, because as a high throughput studies, we also don't want to spend too much time just taking the images, but also still not compromising on the quality. So the trade-off that we did was to have like a sweet spot where we can still get enough quality of images that we can use to do with the pipeline that we then created. But biologically, I think, no.
Okay, good. That's good news. Regarding the variability when you are producing your models, for example, also in the picture you showed in the presentation, where you have multiple systems within the same microwell, did you notice that by scaling down the size of these aggregates, variability was increasing because a small amount of cells were more subjected maybe to stochastic changes? Or would you have had the same variability also in regular non-downscaled models?
You can do 1 or 2 sentences, if you want.
Sorry, I wanted to clarify on the question again. Is it?
Yeah. So you said, so you asked me, yeah? Stochastic.
Okay, can you?
Was variability a concern in your case within the same microwell?
Yeah, at least with the model that we have currently, no, we were able to achieve an efficiency of 80 to 85% in most of the studies, at least in the control scenario. So, no.
Thank you.
So more during the reception. Here we are. Dear Mrs. Candidate, the time appointed for your defending your thesis has passed. The degree committee will now withdraw to discuss the quality of your thesis and your defense. And I request for you that you and your company await the results of our deliberations in this room and wait on our return.
standing right here.
Vinidhra Shankar. The degree committee here present has assessed the quality of your thesis and your defense. In view of its positive verdict and taking into account your previous qualifications, the degree committee has decided to grant you the degree of doctor. Dr. Gieselbrecht is authorized to confer upon you this academic distinction in accordance with Dutch university custom, but before he does that, he will take a declaration of integrity from you, and you have to agree with that. I invite your supervisor to now take the floor.
Please all rise.
Vinidhra, do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent, and responsible?
Yes, I promise.
By the authority vested in us by law and in conformity with the decision of the committee here present, I hereby confer upon you, Vinidhra Shankar, the degree of doctor and grant you all rights attached by custom and law. As evidence of this, I now present you with a degree certificate signed by the rector, the secretary, and the other members of the committee, and affixed with the official seal of the university. Ladies and gentlemen, esteemed colleagues, friends, and family of our newly appointed doctor. Today we have come together to share and celebrate a truly remarkable achievement, the successful defense of Vinidhra's doctoral thesis. After several years of very hard work, this is an important milestone in your academic career, but also in your personal journey. First of all, let me say you how genuinely happy I am for you and congratulate you on this achievement. But maybe wait, let's start at the beginning and take a moment to look back on this incredible journey. After completing your master's thesis at TU Dresden on the role of thyroid hormone signaling in zebrafish brain regeneration, Vinidhra was looking for new challenges and opportunities, and she applied for one of the first PhD positions in our project on embryo models, funded by the local foundation Stichting de Bijenhorst, and she impressed us with her genuine interest in the topic from the very first interviews. We offered her the position, and so she began her PhD in September 2019. And yes, the timing of her start had its consequences, as it coincided almost exactly with the beginning of the pandemic. So her start was a bit rougher than expected. She began her PhD when COVID-19 actually hit the country and basically everything changed. And yes, we all know what that period was like, but for her, it was of course hard because it fell right in those first months when you usually need time to settle in and find your direction. So lab access was reduced to a minimum, personal meetings were very limited, many things were uncertain. And I remember in our very first discussions, when also the scientific ideas in the new project were still a bit vague, Vinidhra was understandably nervous about how to start and what would be expected from her. And that's something you could literally see on her face. I also remember very well that she audio recorded some of our first meetings, and at that time I was not entirely sure what to think of it. And as a PI, you suddenly become very aware of what you're saying. But knowing her now, I'm sure that her intention was simply not to miss even the smallest detail in our conversation, so she could perfectly prepare and be ready for the practical work in the lab. Thanks to the well-organized contingency plans at Merlin, Vinidhra was soon able to start, at least to do some experiments in the lab. And like many others at that time, we also considered alternative approaches when lab work was not possible. For example, writing a scientific review. Step by step, she found her way into the project, into her own research line. And I bring this up because I'm still very impressed by how much she has grown from a slightly insecure PhD newbie into an independent and confident scientist. And as you just saw in her presentation, Vinidhra worked on upscaling assays, on screening approaches to automatically identify particular morphologies and traits in her models to better understand embryonic development or the effects of environmental factors on development. She used computational tools to categorize the identified hits into different classes. So if we were to use a similar analytical approach on Vinidhra herself to better understand who she is, the first results from such an analysis would be quite straightforward. So we would classify her as a human being with a height of roughly 180 centimeters, give or take a few centimeters, and a total volume of around 65 liters, consisting of roughly 40 trillion cells. But as she knows very well from her own research, such basic numbers do not tell us much about the full phenotype or about the traits that really matter. For this, we need to have a closer look at the categories and patterns that appear over time after several runs of screening. On a more visible level, one of the most characteristic traits of Vinidhra is her consistently positive attitude. So whenever she walks into a room, she's smiling, often laughing, she spreads a lot of positive energy around her, like in this very moment. She's kind, very friendly, and always willing to help. This has made her a very pleasant colleague, and she was one of the key elements why we had such an excellent atmosphere in our team over the years. Another early observation that showed up on the screen concerns her eating habits. I remember that she once told me that chili flakes appeared to be a constant component of all, so literally all her meals, at different times of the day. Something that clearly stands out from typical Western European eating habits, I would say. However, I'm not sure if this is still the case, and maybe more recent data might show that this specific trait is no longer significantly expressed. Maybe something to be tested at the reception afterwards. So speaking of changes, we also observed some interesting plasticity over time. In the beginning of her PhD thesis, it was quite difficult to schedule meetings before 10 o'clock in the morning. The problem was that I'm someone who likes to start the day early, while she obviously preferred a slightly later start. So as you can imagine, our ideas about scheduling didn't always align perfectly. But I believe in the end, we always found a way that worked for both of us. But interestingly, I recently noticed that also meetings before 10 o'clock have become more and more possible. So this might be due to a kind of epigenetic effect, where the switch to a more industrial environment might have influenced the expression of certain traits. A very strong and steady category in her profile is for sure her motivation and work attitude. Here we have a lot of data points and many observations during her PhD confirm that she's always highly motivated, very dedicated, and strongly goal-oriented. She's very curious and has a real enthusiasm for scientific questions. Because of her hard work, her knowledge, and her excellent practical skills in the lab, she has repeatedly shown that she can reach her demanding and ambitious goals. Another important and striking trait which can be distilled from the Vinidhra screening data set is her interaction with other people. Supervisors, colleagues, collaborators, interns, and students. Vinidhra's positive attitude and openness make her an excellent team player, and she's an outstanding supervisor and trainer. Together with our intern Nasia, who is probably also here today, yep, indeed, she built a very productive team, or I would even say kind of a well-functioning and a well-oiled screening unit to run the large screening experiments. Their collaboration was so successful that, as far as I know, Nasia decided also to stay in the Netherlands. They become close friends, And I heard that now they are even neighbors. So this is just one example, but I think this says a lot. Her teamwork extends well beyond the lab. Vinidhra is a very social person. She enjoys spending time with colleagues also after work and often takes part in planning and organizing activities. She has been very active in the wider PhD community at Maastricht University, playing an important role in the PhD Academy and eventually becoming its president. This requires time, energy, and commitment, which she was always willing to invest alongside her scientific work. Another clear outcome and important category of the Vinidhra screening is her scientific qualities. And her scientific track record speaks for itself and clearly demonstrates her abilities. With today's defense, she adds another strong data point to an already impressive scientific landscape, and with high-quality publications and solid contributions over the years. She has grown into an independent and confident scientist, and I'm very proud to see how much she has developed during her PhD. And it has been a real pleasure to accompany you on this journey. Now, as we're all here to celebrate this great achievement, I would like, on behalf of the entire supervisor team, Erik, Clemens, and myself, to thank also the assessment and defense committee. Your time, feedback, and critical input are very valuable, and we're grateful for your efforts. And finally, dear Vinidhra, or to be more precise in our scientific analysis, Dr. Shankar, our warmest congratulations. Your PhD journey started under difficult conditions, but you overcame all challenges with determination, perseverance, and your very special positive spirit. The traits and skills that I have just classified, your motivation, kindness, teamwork, curiosity, and resilience, together with your scientific expertise and skills, will be major assets also in the years to come. And we're more than happy that you decided to stay around and keep working together with us. Congratulations also to your family, who traveled the very long way to Maastricht to be here with you today and to share this important moment. We're all proud of you. And with that, I would like to return to the floor to the director.
Dear Dr. Shankar, also on behalf of Maastricht University, I congratulate you, but especially also your Amma and Appa for the degree you have acquired. Before I conclude this session, I have two housekeeping messages. One, there will be a reception on your costs, I presume, in the rafter, which is the previous dinner hall of the monks that lived here in this former monastery. And if you just follow, if you just see drinks, then you follow that direction. One of my colleagues said after a dissertation, not the hora est, but horeca est. And the second housekeeping is we will make a big picture of you and this committee, and then we make a second picture at the same cost of you and your family at the bottom of the stairs of this in the hall. But the remainder of the public can also go to this rafter and already enjoy some hydration fluids. And with this, I conclude this academic ceremony.